That's the hardest problem of all. So, my name's Gary Beauchamp. I have been at Monell for more than 50 years and was here virtually at its founding. And I have loved my life here, and I still come in almost every day, and I'm still writing papers, and I'm still fascinated by the field, and I love the place, and I love what I'm doing. I was a graduate student at the University of Chicago and was looking for a postdoc or some sort of job, and I got an offer from this very small institute at the University of Pennsylvania. I came out, and the director at the time was recruiting young people just finishing their PhDs in supposedly a training position, but of course it wasn't really a training position because there was nobody to train. We were on our own, and what I saw of the place is, this is a place I can go for a year or two, do whatever I want to do, hopefully get a few publications, and then get a real job. And it turned out that I never got a real job. And it's been a fantastic place to work and a family place to work. It felt like we were all together, all as getting salaries of about $6,000 a year, which is what I started at. But we didn't pay any attention to that because we were really having so much fun with science. And some of the things I started doing then carried on from what I'd done as a graduate student, on looking at how animals communicate by smells, looking at how they maybe signal other things like disease, how they're attracted to each other by smell. And I spread that out into studies with infants and children, and began to get my own postdocs. This is the only place on earth that had postdocs who had postdocs. Those people have gone on to be fantastic scientists, and that's one of the great things, too, about being in academia is you're creating a family of children who are not biological children, but they're psychological children. And I am so proud of all of them. And certainly my scientific career, such as it is dependent totally on those people. Everything that I've done is supposedly published and have written about is really, really due to the people that worked with me. And that's true for a lot of scientists. Some will admit it, some won't. I certainly admit it and, in fact, celebrate it. Currently, my technical position is distinguished member. I also go by a director and president emeritus. But mostly I'm just a plain old scientist right now. I was director of the Institute for 24 years. So before that, I was a regular faculty member, first a postdoc, then a regular faculty member. Then I moved into some administrative roles, and then when my predecessor unexpectedly died, there was an attempt that we were going to look for a replacement internationally. And one day, as I was writing up the description of what the job would be to help find this position, two people walked into my office. One was a founder of Monell, a man named Hank Walter, who had been president of and CEO of International Flavors and Fragrances. And he was really a dynamic guy, and I'd gotten to know him over the years. And he was a strong supporter of the idea that the world needed an institute that studied smell and taste. Now, it's because he was in business in a company that sold smells and tastes, namely this International Flavors and Fragrances. But he was really interested in the science. The other person that came in to the office was a man named Lou Thomas. Lou Thomas was at that time president of Sloan Kettering Cancer Center, and I had been working sort of with him on a scientific project for a couple of years. He was an amazing man. He wrote one of the most famous popular science books in the world. Even now is considered one of the great ones, called Lives of a Cell. He was a cancer researcher. So they came into the office and they said, we're not going to search for a new director. You're it. So I never applied for the job. I said particularly to Thomas, because he was the real academic person, full professor, of course, but director of Sloan Kettering. I said, well, I'll do it under one condition, and that is I can spend half my time being a scientist. And he said, and I'm not sure he was right, but what he said was, I wouldn't have wanted you if that wasn't what you wanted. And so I've been able to do this. Probably not such a great administrator, probably not such a great scientist for that matter, but it's been a wonderful trip, and the reason I'm still here is because there's no place I'd rather be. So I was working with a professor, chairman of the department at the University of Chicago, and he studied imprinting. He was a student of Konrad Lorenz, who was Nobel Prize winner in imprinting. And my thesis advisor was brilliant, but he had two kinds of graduate students. One that basically played the role of a son. He bought cigars for him. He drove him around. He did all that kind of stuff. I didn't do that. What I did was, he said, here's a bunch of space. They'll take it away from me if I don't take it up. So I basically had a small building to myself. He studied imprinting, which is the original finding of Lorenz, that birds that are exposed very early in life to some object, maybe another bird, of course, in real nature, but it could be even a different species, will be attracted to that, and that effect will last forever. So I tried to do some of that kind of work with mammals, which nobody had done before, in particular guinea pigs, because guinea pigs, like baby chickens, when they're born, they're really ready to go. They're fully formed. And so they had the same kind of thing as chickens. When they hatch, obviously they start running around right away and they become attached to their mothers. So I did that with guinea pigs, and that's what I did my dissertation on. Then I came here and brought my interest in the smells, which is what guinea pigs use. Chickens use their vision, guinea pigs use their smell, and looked at how animals communicate by smell. And that was what I did when I came here, and was given essentially the same thing. I was told by Morley Kerr, who was the director at the time, here's a room, here's a few $1,000s, do something. And the other thing that was really interesting about those times was very, very little research work was being done about this kind of thing, particularly with mammals. And the director at the time hired not only me and a couple of other people to do kinds of things I do, but also chemists. Those were the founding ideas about the Institute. We had chemistry and biology, and we brought those together in ways that, at least in the field that we were in, had never been done before. And that has been one of the signal research areas in this institute since 1970, which is when it really started. And we have led the world in doing that kind of work. And I'm very proud of that. I was given this room, and I was given the opportunity to. Actually brought some guinea pigs with me from Chicago, and set up. And because we were interested, or I was interested, in what animals can learn about each other, kind of like if you think of what you can learn about a person by looking at him or her, what does an animal learn about another animal by smelling him or her? And so I started looking at made the incredible discovery that male guinea pigs like the smell of females. They're highly attracted to them. And I developed an assay to demonstrate this. And I got a chemist who was working with us at the same level I was. He was a postdoc from Switzerland. And we decided we would do some studies to try to identify the chemistry of how the male guinea pig was attracted to the odor of females. And I worked on that problem for 25 years, never solved it. Very, very hard chemistry problem. But we did a lot of interesting things with it and demonstrated all kinds of important roles that smells have in animal communication. One of the most interesting ones was ones that were done along with my old colleague, Lou Thomas, where Thomas made the suggestion that it was well known that dogs, for example, can tell individuals by their smell. So basically, this has been used in police work and all kinds of things, but nobody really understood how the smells were produced and what the nature of these smells are and what they are able to signal. But he had a really great idea, and this is a thing that makes science so neat. He was an immunologist, and his idea was that there's a set of genes that all vertebrates, and even broader than that, but all vertebrates have, which are called the major histocompatibility complex genes. And these genes are ones that code for your immune system. Essentially, they code for the identity of your immune system, and they are the ones that we were at the time understood to be ones that you had to match in order to do transplants. So that every human being was, aside from identical twins, was different at this set of genes. So wouldn't that be an interesting idea for maybe those same genes would be involved in coding each individual with a unique smell? So that was his idea. And basically what he put it in typically great words, that if, for example, I needed a kidney transplant, it ought to be able to get a dog to smell me and then smell 100 other people and find the one that's most like me and that would be the best donor for my kidney. Crazy idea. And then I spent 15 or 20 years studying that. And that's true. We demonstrated it in many, many different ways. One of the most interesting ones is that when we could take we did this with mice mainly. Guinea pigs I gave up on, too big. They're too big. We had to use mice. If you take a female guinea pig and mate it with a male and then collect the urine from the pregnant female, you could identify who the male was. That is, the pregnant female signaled who she had mated with, and that fetus then signaled out to the outside world who the father was going to be. And this, we think, plays a big role in mate selection in mice, of course. We have evidence similarly in humans, and many, many species maybe underlie this. This is one of the ways that people organize themselves and may account for some of the mate choice amongst humans and some of the avoidance of mating or marrying or whatever between very close relatives. So really neat stuff. And again, one of the things that's been great for me and has been great for many of the people that are here at Monell is one develops a series of research projects that expand and expand and expand. And this kind of work actually has been expanding into identification of disease by smell, because just like the individual identity, diseases produce different odors, and we believe that we will be able to, and we know we can actually identify some diseases based on volatile smell molecules. People were brought in, and then I started bringing them in myself, who were compatible with my interests. And so the great thing about that is they ended up helping me by doing what I was interested in doing, and I think helped them, get into a field that was worthwhile and made careers for them, for many of them. And so it was mutually supportive. And it was particularly lucky for me, I think, that I was here at the beginning, and so had my fingers in before almost anybody else. And so many of the things that I did was based on finding people that I thought were interesting, that were doing interesting things, hiring them as my postdocs or graduate students or what have you, and at least for the early years, making them then colleagues, and then they taught me. Because for the most part, what I wanted people was new stuff that I didn't know, and that was of great benefit. Just think of one person. It's a woman named Julie Monell. Julie came to work with me as a postdoc, and interestingly, she came from the University of Chicago program that I did, same program. She studied animals there, but by that time I was doing a lot of work on humans, and particularly in babies, and how they detect smells of food and attraction to their mothers and things of that sort. She wanted to learn about human work, and I wanted somebody who knew a lot more than I did about a lot of things about human biology and infancy and such like. And so she did a couple of experiments, which I think I had in the back of my mind, but had never really thought that I could do during her first couple of years as a postdoc. And then, of course, took off, and she became the world's expert in this area. And I learned a lot from her. One of the funny things about this is that she and I just won a famous award, the Ig Nobel Award, which is an award given out sort of mimicked on the Nobel Prize awards, but it's for work that was done that makes you laugh and then makes you think. And basically what she did was to show that if you took mothers and fed them flavors, and in particular we used garlic and some other flavors, that their milk would take on the odor of these flavors, and even the amniotic fluid would take on the odor of these flavors, and that resulted in the babies imprinting onto these odors. And when they were born, then they loved those flavors that they had been exposed to in utero, and they loved them for the rest of their life, as far as we can tell. And so there's my going back to guinea pigs and coming to people. That paper was published in 1991, which is a long time ago now. But that has set the stage for a tremendous amount of work, again, by many, many other people. And that's the other thing that really emphasizes. One of the things that scientists measure science by is citations. That is, you publish a paper and you hope somebody will read it, and then you hope somebody will use it to advance the fields. And in my life, I've had a lot of citations, but almost never are my citations my work alone. They're always somebody else, and they're mainly the senior person really was the other person. And Julie was an example of that. I've got a whole bunch of other examples like that, where the basis for something maybe comes out of some work we did at one point, and that bludgeons out into just massive things. Well, by far what I care most about is the people I worked with and their success. There's just no question about that. But in terms of the specific science part, I have to take some examples of it. And one example, which I've always loved, we had a lot of activities with companies, and one of the companies that we worked with was a pet food company. Working with them, I had the chance to study what cats like to eat. And it seems so naive now, but at the time, the feeling was that every animal liked sweet things. So we, thinking that that was true, decided we wanted to test cats, how sensitive they were to sweet things. And so we did this first with domestic cats at Monell Center, and then with cats at the Philadelphia Zoo, which happens to be very close to where we are. So we looked at lions and tigers and leopards and jaguars. And lo and behold, we found, as far as we could tell, they didn't like sweet at all. It's not they disliked it. It seemed like they couldn't even taste it. And so we published that paper a long time ago, in the 1970s. We've got a lot of pushback from some people who claim that cats do like sweet taste. But I always stuck with my guns and said, well, we couldn't repeat that. So years later, working with other postdocs, we decided to try to find out how it is that other species, like mice and humans, like sweet things so much. And it turns out, this is so common in science, we and 4 other groups at essentially the exact same time, around 1999, 2000, identified the specific receptor in people's tongues that detects sugars. And as I say, it was detected at the same time by 4 or 5 different groups. It's because of progress in molecular biology it was able to be done. But once we did that, then I went back to my cat work, and I said, wait a minute, let's look to see whether cats have those same receptors. And another colleague, a very close colleague of mine who did molecular biology, and a colleague from Russia who had been a postdoc and became a very productive scientist here, looked at cats, and lo and behold, it turned out that cats and many other species now we know that eat only meat and don't eat, say, sugar, they don't eat plants, they've lost the ability to taste sweet. Basically, the molecular receptor that is able to detect the molecule of sucrose or fructose or glucose, any sugar, the gene that codes for that molecule has lost its functionality. And so these animals literally do not live in the sweet world. That work now has been extended all over the place, people looking at different species, mammals, vertebrates, birds, whatever, work that we don't do here at all, never did do here. That initial finding was really one of the most exciting things that we've ever done.